Okay, so let's work through exam 3 together. Okay, so your short answer question number 1 it talks about your cousin being pregnant. So your cousin is pregnant and close to delivery with her second baby, and she seems to have group B strep. And this bacteria is not harmful for your cousin, but it can cause serious infections for the baby when the baby's born. And so the doctor finds that she does have group B strep, and tells your cousins that she will have to have an IV antibiotic administered when she is admitted to the hospital for labor and delivery. Your cousin knows that you understand biology and asks you the questions below. So she wants to know what a group B strep cell looks like. Okay, so this is a gram-positive bacteria. So first we're going to start by drawing out a bacterial cell. So we've got our cell membrane, our circular DNA, I need to add in label. We've got some ribosomes. There's not a nucleus. There is an RNA polymerase here too. And then there's going to be some peptidoglycan, a lot of peptidoglycan since it's gram-positive bacteria. There's a lot of peptidoglycan on the outside. Okay, so that's the basics, right? So we've got RNA polymerase, we've got ribosomes, there's a circular DNA chromosome, and cell membrane, and peptidoglycan. Okay, what could the antibiotic be targeting that to be able to target the strep B bacteria? Okay, so it could be targeting the peptidoglycan. That's a big one, because that's the only found in bacteria, right? It could be targeting simple polymerases, or those simple ribosomes too, right? So anything that isn't going to harm the baby. So it's not going to be harmful for the baby, right? So it won't harm the baby cells at all. Next is whether or not they should have skin-to-skin contact and still breastfeed and all the things. And so, yes, definitely start helping the baby prep the microbiome. Okay, so remember the competitive exclusion principle, where if there's good bacteria, right? So if there's good bacteria there, then when a baby encounters some bad bacteria, it won't be able to harm it. It will just go straight out its system, right? So something about the competitive exclusion principle. And you can also tell them that the microbes are going to, these good microbes will help with digestion and immune response. Oops. I don't know what I was writing first. Immunity and all kinds of other things in addition to the competitive exclusion, okay? Next is that antibiotics are a drug used to kill bacteria and are sometimes necessary at large chicken and turkey farms to keep the flocks healthy. In the past, many companies continuously added antibiotics to the feed, and the antibiotics then ran off the farm and into the surrounding ecosystems. So this is the reason why you see chicken labels that say no antibiotics ever, right? But think about how sometimes we get sick and need antibiotics, and so sometimes chickens might need that too. But this is the reason why that movement kind of started, okay? So here are the questions. What roles do bacteria play in ecosystems, and how would the continuous use of antibiotics at a turkey farm impact the ecosystems around it? So bacteria play many roles, right? Right? They can play many roles. They can be producers, right? We've talked about them being producers. We've talked about them being decomposers, right? And these bacteria can be food, right? for other organisms as well, right? And if we have this continuous use of antibiotics, then we're going to have no bacteria, right? And if we have no bacteria, then they can't play those roles. Okay, some farmers are now feeding their flocks probiotics instead of antibiotics. Why are they doing that? So remember, these probiotics are good bacteria. Maybe I'll add that in the notes. Okay, are good bacteria and will help the turkeys. Or chickens, whatever, fight off the bad bacteria. Right, so this is that competitive exclusion principle again, right? It's also going to help them in the same ways as before. So bacteria, these good bacteria are also going to help with digestion and immunity and other things as well. Okay, could there be a way to incorporate turkeys into an ecosystem without causing harm? So into like natural ecosystems. So you guys could go lots of directions with this. So there are turkeys in nature, right? Nature has turkeys. Right, so nature does have turkeys in it, right? So you can go and see wild turkeys sometimes out in the woodlands, especially. And it is part of the ecosystem. They are part of the food web that's present in natural ecosystems, right? But you could also have farm systems where you have turkeys play a role. So I allowed a lot of different answers for this. So some people were talking about like regenerative farming, and that is great. I'm surprised some of you knew about that. And that's great. So you can do regenerative farming or sustainable farming, right? Some of you talked about that, but there's lots of different ways that you guys could go with this that I allowed. Okay. All right. So the next one, this actually is only 6 points. I'm working off of an old draft. Okay. So put a star of the MRCA of leafwing and seawing. So a star at the most common recent ancestor of leafwing and seawing is right there, right? So that's where we connect the two together. Circle the smallest clade that includes nightwing. Okay, where's nightwing? Nightwing and sandwing, okay? So you need to make sure that you included all of them, including the most recent common ancestor, okay? So the clade includes that common ancestor as well. Could the most recent common ancestor of all dragons breathe fire? So it looks like fire breathing is a this is breathing as a defense strategy started here, and then fire breathing started there. So only these 1, 2, 3, 4 dragons are going to be able to breathe fire. So no, fire breathing is a synapomorphy for just 4 species. Okay. Who is more closely related, icewing and seawing? Okay. So icewing and seawing, or icewing and skywing. Okay. So here, icewing and seawing, their most recent common ancestor would have been here, versus icewing and skywing, their most recent common ancestor is here. So I'm going to choose this one. And the reason is they have a more recent common ancestor. Okay. All right, so question 4, tree A shows the correct relationships between 5 species. Okay, so A and B are most closely related, D and E are really closely related, and then C is closer to D and E than it is to A and B. Okay, which of the phylogenetic trees, B or C, shows an evolutionary relationship that is different from the other two? Okay, so A and B are shared here, and then C and E are different here. Okay, so if D and E should be closely related, and here it says C and E, then this is the difference, right? Okay, let's check quick with tree C and see if it matches. So A and B is good, D and E is good, C being closely related to D and E is good. So tree C is the same as A. So tree B is different. And that's because it shows C and E being closer. But tree A says that D and E are closer. Okay, so the next thing I need to do is fill in tree D to show the correct relationship shown in tree A that differs from the trees already shown below. Okay, so here I need to take a look at my tree, and it looks like here's where my trio is, where I have C, D, and E, and then on this other side I'm going to have A and B. So A and B is most closely related, D and E are really closely related, same as A and B are to each other, and then C is really close to D and E. Okay. All right, so the next one is to use the character matrix to the right to create a phylogeny and include all species and all synapomorphies. So note that it's worth 15 points. I thought it would take you guys a little bit of time, right? So here I'm going to start grouping things together, and I want to find things that have just like two things that are grouping things together. So it looks like the cardinal and the human are both tetrapods, and then the insect and the butterfly are both insects. The bumblebee and the butterfly are both insects. The redbud tree and the bean plant are both legumes. And the mushrooms and the bread yeast are both have chitin in them, okay? All right, so the next thing I want to look at is to see if there's anything that groups three of them together. So I'm just going to change my pen color just so that I can kind of see what is going together. So I have the redbud tree, the blackberry, and the bean are autotrophs. And then I have the cardinal, the human, the bee, and the butterfly are all animals. And then I've got some heterotrophs here too. And that doesn't really tell me a lot because that's just like the opposite of the autotrophs. Okay, but maybe it'll help me group two groups together. So I'm just going to start somewhere. I'm going to start, let's start with the mushrooms because they're up top. So I've got the morels and the bread yeast. So I've got the fungi together, and chitin is the thing that grouped them together. And then I had lots of heterotrophs there, including a bunch of animals. So let's go to the animals next. So I'm going to just change color so it's easier to see. So I've got the cardinal and the human are both tetrapods, right? And then the other animals were the bee and the butterfly, and they were more closely related to each other here too. So the bee and the butterfly are both insects. And then those were all grouped together because they're all heterotrophs. Oh, wait, they're all grouped together with, the fungi because they're all heterotrophs. That's what I was wanting to do. Oops. Why won't my pen go back? Okay. They're grouped there because they're all hets. These up here are all animals. Okay, and then let's see where I'm at. So I've got the cardinal, the human, the bumblebee, the butterfly, the bread yeast, and the mushroom, the morel. And so now I've got my autotrophs, I've got my plants, and it looks like the redbud tree and the bean plant are more closely related to each other. So I'm going to do a redbud and the bean plant, and then the blackberry bush is outside of them. So they were together because they're legumes. And then we have the blackberry. Right? And they were all plants, they're all autotrophs. In this case, and plants. I'm just gonna put that in parentheses. Okay. All right, and then all of these are eukaryotes. Okay, so we've got our full tree. All right. So I think that's it. So we've got our red bud, we've got our bean, we've got our blackberry bush. We added heterotrophs and autotrophs. We have animals, tetrapods, insects, legumes, and the chitin. Okay, next question. So question 6 is to label this tree. So I've got archaea, gram-positive, gram-negative bacteria, and eukarya. And then I've got some synapomorphies that I need to add on too. So let's start by seeing what the tree has. So the tree already has mitochondria, so I know that that must be eukaryotes. And I know that, or eukarya, either is fine to write there. And I know that they also have a nucleus. So I'm going to add that there. And then their closest is going to be the archaea because they share things like complex ribosomes. So I'm just going to knock that off and that. And I'm going to add complex ribosomes. And then I've got gram-positive and gram-negative bacteria. So gram-positive and gram-negative bacteria. And they both have peptidoglycan. Okay, and then all of them have DNA. Okay. Next is drawing out an archaea cell and a protist cell. Okay, so remember that protist is a eukaryote, right? Okay, so we're going to label both cells with different elements that we learned about. Things to include are circular and linear DNA, mitochondria, cell membranes, peptidoglycan, ribosomes, polymerases, and the nucleus. Okay, so let's draw out the archaea first. So here's the archaea, and it's going to have circular DNA in it. It's going to have complex ribosomes. It won't have any mitochondria. It does have that cell membrane that I already drew. It does not have any peptidoglycan. Neither of these have peptidoglycan. It's going to have RNA polymerase and DNA polymerase on them, maybe if it's being copied, but no nucleus. And then the protist is going to have a nucleus, and it will have linear DNA in it. And it will have that complex ribosome again. It'll have those polymerases. It has that nucleus. I'm going to draw the endoplasmic reticulum because I can't not draw it. Okay. And the mitochondria does have. So there's an inner and outer membrane. It has circular DNA on the inside of that. Okay, so I'm going to label and then see if I can check everything off. So I've got complex polymerases in both. I have complex ribosomes in both. I have circular DNA here in my archaea, and I also have it here in the mitochondria. MT, so mitochondria with circular DNA inside of that. Here I have my nucleus with linear DNA. Okay, so now let's go through and check. Okay, so I've got all circular and linear DNA in the cell. I've got the mitochondria only in the protist. Oh, I need to label my cell membrane. I've got complex ribosomes and complex polymerases, and then nucleus only in the protist. Okay. All right, I think that's it. So below is a eukaryotic cell. So label the nucleus and describe if the DNA within is linear or circular, and describe how the, okay, so let's just do that first. Okay, so label the nucleus. So this is a nucleus. And the DNA inside is going to be linear. The nucleus or an endoplasmic reticulum arose, remember, from infolding the inner and outer membranes of the mitochondria. So this is the outer, and it's eukaryotic, or even if you put like archaeal, I accepted that too, archaea. The inner is going to be from bacteria. And then here we have circular DNA. Okay. And then next it wants me, okay, so I did that. Next it wants me to draw what will happen next with photosynthetic bacteria during endosymbiosis and afterwards. So it's gonna eat it. So you could do lots of things. You could have gone like this to encircle it, or drawn it all the way out from the start, where you did something like something like this, where you said, okay, it's gonna go like that, right? So your nucleus and your ER, mitochondria. And then afterwards, it's going to be inside. So nucleus, ER, you have your mitochondria, and then you have your photosynthetic bacteria, and it's going to have circular DNA, just like the mitochondria. So what type of DNA will be in the photosynthetic bacteria? It's going to be circular, right? So this is going to have that circular DNA. And this becomes the chloroplast. Right? Okay. Now, just keep in mind that the chloroplast doesn't look exactly like it looked when it was first engulfed, right? It has evolved since then, too. So just kind of keep that in mind for the future. Okay, so next is to label the tree below with the following, and you can use each one more than once. Okay, so I need to first figure out what I have. I've got animal, archaea, fungi, plant, and protist. And I can start looking for synapomorphies that are on my tree of things that I know. So I see that the chitin is there first, so I know that this must be fungi. I know that the animals are really closely related to the fungi, so they must be over here. This right here was kind of hoping you guys would put a protist, but if you had all of this as animal, I was also okay with that. Next, I need to have the plants. So here I have the plants that have their roots. Here's another protist, photosynthetic protist, more protists. Okay. And then I did want you guys to use each one at least once. So over here is archaea. Okay. Next, you need to be able to look back at semi-conservative replication, because that's how binary fission happens when we replicate those prokaryotic cells, right? It still happens through semi-conservative replication. So the first thing I wanted you to do was to label the 3' and 5' ends. Most of you guys got this. And then replicate the DNA, and most of you guys could do this too. So you did 5', A, C, and some of you even did this, which I thought was great, where you just took this and copied it. Oh, I don't know how you guys did that. Okay, I'm just going to write it out. Okay, so 5', A, C, T, T, G, C, A, T, G, C, T, A, G, T, C, T, C, GA 3 prime. Okay. And then, and I think I shortened this for you guys because it's really long. Okay. T, 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 G, A, C, G, T, A, C, G, A, T, C, A, G, A, G, C, T. Okay, hopefully I didn't mess any of that up. If you guys had like one typo in there, I was fine with it. And then I'm just going to put my new DNA in a different color so it's easier for me to track. And I wrote that in the wrong direction. Would have been better if I wrote 5' to 3', like polymerase, but I didn't. Okay. And here I did want to see that you guys knew that you had to still use the T's because you're still using in DNA, right? Okay. So I've added those 3' and 5' ends. I've replicated. Oh, and then specify which are old and which are new. Black are old. And the blue are new. So are the two DNA sequences, just the sequences themselves, identical? Yes. They were made by using each strand as a template. So this old strand, this old strand matches this new strand, and this new strand matches that old strand, right? If you look at them, they're going to be those exact same sequences put together. Okay, what types of bonds had to break to replicate that DNA? Hydrogen. What type of bonds hold the two DNA strands together? Hydrogen again, right? So the hydrogen bonds are the ones that hold them together, and those are the ones that had to break to replicate the DNA. Does this type of replication happen only in eukaryotes, only in prokaryotes, or in all cells? So this is all cells, right? This is how DNA is always replicated. Okay, and that's it. That's the end of the exam. I hope, I know I used the old version, so sorry about that, guys. But I think there's very few changes from mine to yours. So I hope that this helps and helps you understand anywhere that you might have missed any points. And I look forward to seeing you guys later this week as we dive into new content.